Very good morning. So today's class is on chromosomes. So chromosomes, what are chromosomes? So you all know what is the highly condensed form of chromatin, right? So we have the nucleus, and the nucleus, there is the genetic material, right? And our genetic material is DNA, and the DNA is packed into chromosomes, right? And it is the chromosomes are the deeply stained, highly condensed form of chromatin. And these chromosomes are made of many chromatin threads, each containing nucleic acids and protein found in the nucleus of most living cells, and it carries the genetic information in the form of genes, right? Chromosomes are made of 50% proteins and 50% DNA. Each chromosome contains many genes that act as a blueprint or set of instructions for the cell. In metaphase, in metaphase stage only we can study about the chromosome, and most of the chromosome studies we have done it in the light microscope only. Okay. In metaphase, chromosomes are divided longitudinally into two sister chromatids, and duplicated condensed chromosomes are known as dyads. The duplicated chromosomes are held together at the region of centromere. So, this is a dyad, and this is the centromere, the constructed part. That is the centromere, and these are the sister chromatids, and these are the non-sister chromatids. And the short arm is called P arm, and the long arm is called the Q arm. And this is the homologous pair of chromosomes. Okay, so in metaphase, chromosomes are divided longitudinally into two sister chromatids, and duplicated condensed chromosomes are known as dyads. The duplicated chromosome, as in the figure, you have seen that in the constructed part, that is the centromere. And typically, a chromosome is made of two chromatids, a centromere, and a secondary construction also. The two chromatids of one homologous chromosome with respect to those of the other homologs are called non-sister chromatids. So there is sister chromatid, there is centromere, there is secondary construction, and there is a non-sister chromatids. And the shorter arm of the two arms of the chromosomes extending from the centromere is called the P arm, and the longer arm is called the Q arm. So P arm, Q arm, then these are the non-sister chromatids, and these are the sister chromatids, and this is the centromere. Okay. So when coming to the classification of chromosome, based on the position of the centromere, we can classify it in different forms. Okay. So we have the telocentric chromosome, acrocentric chromosome, submetacentric chromosome, and the metacentric chromosome. In the metacentric chromosome, you can see the centromere, it is in the middle part only. Okay. And in the submetacentric chromosome, it is somewhat closer to the middle part, right? And in the acrocentric chromosome, it is somewhat towards the terminal part. It is close to the terminal part. It is more close to the terminal part. And in the telocentric chromosome, it is in the terminal part. Okay, that is the telocentric chromosome. So according to the position of the centromere, we can classify it into metacentric, submetacentric, acrocentric, and telocentric chromosome. And what is the chemical composition of these chromosomes? We know that is the DNA and the proteins, and the proteins are the histone proteins. So the packing of the DNA is due to the histone proteins, and it is composed of thin chromatin threads called chromatin fibers. And the interphase chromatin consists of 30 to 40% DNA, 50 to 65% protein, and 1 to 10% RNA. And the metaphase chromosome, it contains 15 to 20% DNA, 10 to 15% RNA, and 65 to 75% protein. So in metaphase chromosome, the protein content is more. And the DNA, it consists of nucleotides, while the primary protein component is histones. Histones strongly attracted to the negatively charged phosphate groups of the DNA. And you know, DNA, it is double helical structure. We have studied this in +2 also, right? So that's why there are 5 major types of histones, thousands of other types of proteins involved in DNA replication and gene expression. So can you just explain me about chromosome? Chromosome, they are the deeply stained, highly condensed form of chromatin, and it is made up of chromatin threads each containing nucleic acid and proteins, and it carries the genetic information in the form of genes, and it consists of 50% proteins and 50% DNA, and the proteins are histone proteins, and it is the blueprint or set of instruction for the cell. And in metaphasic stage, we are studying about the chromosome, and the chromosome, it consists of the centromere, the sister chromatids, and the non-sister chromatids, and there is a secondary construction also, and the short arm is called the P arm, and the longer arm is known as the Q arm. And based on the position of the centromere, we are classifying it into metacentric, submetacentric, acrocentric, telocentric, right? Then coming to the karyotyping. Karyotyping, what is karyotyping? Karyotyping is the process of pairing and ordering all the chromosomes of an organism, thus providing a genome-wide image of an individual's chromosome. So we can pair the chromosome of an organism, and we can find out which organism is this. Okay, so that is the process of pairing the chromosomes of an organism. And karyotypes are prepared from mitotic cells which are frozen in metaphasic stage. So in the metaphasic stage only, we are studying about the chromosomes. From the mitotic cells, we are collecting the chromosomes. And characteristic structural feature for each chromosome are revealed. It can reveal changes in chromosome numbers linked to conditions such as, so we can study whether there are some genetic disorders or something like that. Okay, Down syndrome. Careful analysis can show more subtle changes as chromosomal deletions, duplications, translocations, or inversions. Increasing use of karyotyping for diagnosis of specific birth defects and genetic disorders. So nowadays we have from the birth only we can, in the womb only we can diagnose whether there is some kind of genetic disorders or something by this karyotyping. Okay, so it is the process of pairing the chromosomes of an organism. And how this karyotyping is done? What is the procedure for this from karyotyping? So we are collecting this 5 ml of venous blood and adding the phytoheme agglutinin. Okay, phytoheme agglutinin, and placing it in the culture medium, and culture at 37 degrees Celsius for 3 days. Then add colchicine, which arrests the cell division in the metaphasic stage, and keeping it in hypotonic saline for the fixing of the cells, and spread these cells and it digests with, we are giving the proper staining and analyze the metaphasic spread of the chromosome, and we can find it out. We can order the chromosome, we can pair the chromosome, and we can find it out, find out the organism towards the chromosome belongs. Okay, whether there is any chromosomal abnormalities we can find out. Okay, so that is the procedure of karyotyping. Then from the karyotyping, we can find out the chromosomal abnormalities like Down syndrome, Klinefelter syndrome, Turner syndrome. Down syndrome means we have on the 21st chromosome, we have the trisomy. So trisomy 21. It has actually 2 chromosomes. So, yeah, 1 pair of chromosomes, but person has 3 chromosomes, 21st instead of 2. Okay, and there are so many symptoms for this Down syndrome. Later we will discuss on that. And Klinefelter syndrome, it has 1 extra X chromosome, and in the Turner syndrome, they are missing 1 of the X chromosome. So from the karyotyping, we can find out whether it is Down syndrome, Klinefelter syndrome, or Turner syndrome. And coming to the chromosomal abnormality, a chromosome anomaly or abnormality or aberration reflects on a typical number of chromosomes or a structural abnormality in one or more chromosomes. Okay, that is the chromosomal abnormality. Chromosome anomalies usually occur when there is an error in cell division following meiosis or mitosis. There are many types of chromosomal anomalies. They can be organized into numerical and structural anomalies. Numerical anomalies are there, structural anomalies are there, okay? And this is called, where there is abnormal number of chromosomes, it's called aneuploidy, and occurs when an individual is missing either a chromosome from a pair, that is monosomy, or has more than two chromosomes of a pair, that is trisomy or tetrasomy. So aneuploidy means abnormal number of chromosomes. So there are two types of chromosomal abnormalities, that is numerical and structural. And when coming to the numerical, that is when there is abnormal number of chromosomes, we call it as aneuploid. We can see monosomy, trisomy, tetrasomy. Okay, in humans, an example of a condition caused by numerical anomaly is Down syndrome, that we all know, that is trisomy 21. And trisomic has been determined to be a function of late maternal age. And also monosomy we can see in Turner syndrome, where there is absence of one X chromosome. And when coming to the structural anomalies, structural abnormalities of chromosome, when the chromosome structure is altered, this can take several forms. That is, one is deletion. A portion of the chromosome is deleted. That is the a portion of the chromosome is deleted. That means there is deletion. That is in many of the syndromes we can see this. That is, one of the syndrome is Wolff-Hirschhorn syndrome. Wolff-Hirschhorn syndrome, there is partial deletion of the short arm of chromosome 4. So in chromosome 4, there is partial deletion of PR. Okay, and also in Jacobson syndrome, there is terminal 11 Q arm deletion disorder. So you have to see there is numerical abnormalities and structural abnormalities. In numerical abnormalities, we have to write about Down syndrome, Turner syndrome, Klinefelter syndrome. Okay, in structural abnormalities, you have to write about deletion, and for example, that is Wolff-Hirschhorn syndrome, that is a partial deletion of the short arm of chromosome 4, and Jacobson syndrome, that is the 11q deletion disorder. And when coming to the duplication, a portion of the chromosome is duplicated in extragenetic material. That is, it's non-human disorder group. Charcot-Marie-Tooth disease type 1A, which may be caused by duplication of the gene coding peripheral myelin protein 22 on chromosome 17. So it is on duplication on chromosome 17, and its example is Charcot-Marie-Tooth disease type 1A. So a portion of the chromosome is duplicated. A portion of the chromosome is duplicated here. So that is one more structural anomaly, that is the duplication. Then coming to translocation, there are two types of translocation, that is reciprocal translocation and Robertsonian translocation. So in translocation means a portion of one chromosome is transferred to another chromosome. So one portion is transferred to another chromosome. That is there are two types of main types of translocations. That is the reciprocal translocation and the Robertsonian translocation. In reciprocal translocation means segments from two different chromosomes have been exchanged. So it's exchanged from two different chromosomes. But in Robertsonian translocation, an entire chromosome has attached to another at the centromere. Okay, an entire chromosome has attached to another at the centromere. So this is the reciprocal translocation. And Robertsonian translocation is a common form of chromosomal rearrangement that in human occurs in 5 acrocentric chromosome pairs. So it's usually occurs in acrocentric chromosome pairs. So that is the reciprocal translocation and Robertsonian translocation. Next is inversion. A portion of the chromosome has broken off, turned upside down, and reattached. Therefore, the genetic material is inverted and the sequence is disturbed. Okay, so there is the inversion of the chromosomal. What? A portion of the chromosome, it's broken off and turned upside down. That is the inversion, one of the chromosomal abnormalities. Next, coming to insertion. A portion of one chromosome has been deleted from its normal place and inserted into another. That is the insertion. So we have studied the structural abnormalities of chromosome, that is the deletion, duplication, inversion, insertion, translocation. Okay, these all are the structural anomalies of the or abnormalities of the chromosome. And when coming to the numerical abnormalities, you have the Down syndrome, Turner syndrome, Klinefelter syndrome. Okay, so and also we can see the ring chromosome. A portion of a chromosome has broken off and formed a circle or ring. This can happen without loss of genetic material. Okay, we can see a ring chromosomes and also isochromosome. It is formed by mirror image copy of a chromosome segment, including the centromere. So nothing in that. You have to study about the structural anomalies: deletion, duplication, inversion, translocation, and inversion. Okay. Then most chromosome abnormalities occurs as an accident in the egg or sperm, and therefore the anomaly is present in every cell of the body. Some anomalies, however, can happen after conception, resulting in mosaicism, where some cells have the anomaly and some don't. So that is the mosaicism. Okay, so if most chromosome abnormalities, it occurs as an accident in egg or sperm, so you will get in every cell of the body, but in mosaicism, where some cells have the anomaly and some don't. So that's the difference. Okay. In genetics, a mosaic or mosaicism denotes the presence of two or more population of cells with different genotypes in one individual who has developed from a single fertilized egg. The individual is from a single fertilized egg, but there are different two different population of cell in one body. Okay, that's the mosaicism. Chromosome anomalies can be inherited from a parent, or we know that this is why chromosome studies are often performed on parents when a child is found to have an anomaly. Okay, now we have so many genetic studies. When the child is in the womb only, we can find it out whether there is any abnormality. Okay, if the parents do not possess the abnormality, it was not usually inherited. However, it may be transmitted to subsequent generations. De novo mutation, a genetic mutation that neither parent possesses nor transmitted. So, as we have already discussed, we have the Turner syndrome, a chromosomal disorder in which a female is born. We know that female, it's two X chromosome. So, when the female is born with only one X chromosome, that is the Turner syndrome. Okay. And what are the features of Turner syndrome? You can see. Newborn include redundant neck skin and peripheral lymphedema and congenital heart diseases. There is increased risk of most of the adrenal anomalies, then hypertension, autoimmune thyroid disease, obesity. Okay. And when coming to Klinefelter syndrome, that is, it is the most common cause of hypogonadism and male infertility, accounting for about 10% of infertile men. And when coming to the Down syndrome has many features: flattened facial profile, flattened nasal bridge, protruding tongue, dysmorphic facial appearance. Okay, so chromosomes, the DNA, the genetic material DNA is called form the chromosomes, and chromosomes we can study under light microscope in metaphasic stage. We are up and the study of the chromosome, what we can do by karyotyping, we can study the chromosome. We can pair the chromosome and we can find it out which organism it belongs to, and all the genetic disorders we can study. And in the chromosomal abnormalities, we have the structural anomalies and the numerical anomalies, abnormalities, and you have to study about the structural and numerical abnormalities of chromosome. Okay, thank you.